Introduction. Welcome to today's lecture on the fascinating world of cell biology, specifically focusing on the intricate dynamics and functions of microtubules. Microtubules are essential components of the cytoskeleton, serving crucial roles in cell division, intracellular transport, and cell shape maintenance. In this lecture, we will explore various aspects of microtubule biology, including their assembly dynamics, modifications, associated proteins, and their involvement in cellular processes such as neuronal development, cancer, cilia/flagella function, cell migration, intracellular trafficking, and therapeutic applications. So, let us dive into the world of microtubules and unravel their fascinating complexities. Microtubules are dynamic polymers composed of alpha and beta tubulin heterodimers. They exhibit a unique property of dynamic instability, constantly undergoing cycles of polymerization, assembly, and depolymerization, disassembly. At the plus end of a microtubule, incoming tubulin dimers bind more rapidly, promoting growth, while the minus end is relatively stable. This dynamic behavior allows microtubules to rapidly reorganize and participate in various cellular processes. To further augment the functional diversity of microtubules, tubulin subunits undergo various post-translational modifications. These modifications include acetylation, phosphorylation, glycylation, and detyrosination, among others. Each modification regulates microtubule stability, interactions with microtubule-associated proteins, MAPs, and motor proteins, enabling microtubules to perform specific functions in different cellular contexts. Centrosomes play a pivotal role in microtubule organization and cell division. In this section, we will explore the intricate process of centrosome duplication and its regulation. Centrosomes consist of a pair of centrioles surrounded by paracentriolar material, PCM. Their duplication is tightly coordinated with the cell cycle and involves licensing, duplication, and segregation, forming the basis for accurate distribution of microtubules during cell division. Motor proteins, such as kinesins and dyneins, utilize the energy from ATP hydrolysis to move along microtubules, facilitating intracellular transport. Kinesins predominantly move towards the plus end, while dyneins move towards the minus end. These motor proteins play vital roles in diverse cellular processes, including organelle trafficking, mitotic spindle formation, and neuronal transport. MAPs are a diverse group of proteins that interact with microtubules to regulate their stability, organization, and function. They contribute to microtubule bundling, cross-linking, and cargo binding. MAPs such as tau play essential roles in neuronal development and plasticity, while others like MAP4 are involved in mitotic spindle assembly and regulation. Microtubule severing enzymes, such as catenin and spastin, break microtubules into shorter fragments, aiding in their reorganization and participation in diverse cellular processes. Additionally, microtubule remodeling enzymes, including tubulin tyrosine ligase-like, TTLL, family members, catalyze the addition of specialized tubulin isoforms to microtubules, further expanding their functional diversity. In this section, we will explore the crucial role of microtubules in neuronal development and plasticity. Microtubules are essential for axon growth and guidance, dendrite formation, and synaptic plasticity. They provide the structural framework for transporting organelles, proteins, and neurotransmitter receptors, crucial for neuronal function and communication. Aberrations in centrosome structure and function have been implicated in cancer development and progression. We will examine the link between centrosome abnormalities and genomic instability, centrosome amplification, and their impact on mitotic spindle formation, leading to chromosome segregation errors and aneuploidy, hallmarks of cancer. Cilia and flagella are specialized microtubule-based structures protruding from the cell surface. We will discuss the role of microtubules in ciliary and flagellar assembly, beating, and signal transduction. Importantly, defects in microtubule-based motility in cilia/flagella are associated with various genetic disorders, termed ciliopathies. Cell migration is essential for embryogenesis, wound healing, and immune response. Microtubules orchestrate the spatial organization and dynamics of focal adhesions, cytoskeletal rearrangements, and force generation, driving cell movement. We will delve into the molecular mechanisms underlying microtubule involvement in cell migration. Microtubules serve as tracks for intracellular transport of vesicles, organelles, and macromolecular complexes. Molecular motors, such as kinesins and dyneins, transport cargoes along microtubules to precise intracellular destinations. We will explore the intricate coordination of motor proteins, adapters, and regulatory factors in ensuring efficient intracellular trafficking. Lastly, we will discuss the significance of microtubules as targets for therapeutic interventions. Microtubule stabilizing drugs, such as taxanes, and microtubule destabilizing drugs, including vinca alkaloids, have emerged as potent anti-cancer agents. We will delve into their mechanisms of action, clinical applications, and potential side effects. In conclusion, microtubules represent a fascinating area in cell biology, exhibiting remarkable complexity in their dynamics, functions, and regulatory mechanisms. Through their ability to undergo dynamic instability, interact with numerous associated proteins, and participate in diverse cellular processes, microtubules are central to the proper functioning of cells. Understanding their biology and exploring their potential therapeutic applications holds great promise for advancing our knowledge of cell biology and developing novel therapeutic strategies for various diseases.